So topic is microanatomy of intestines earlier, so I won't be speaking much of it. So when I say intestines, what I'm going to speak about is small intestine and large intestine. Have you all dissected? You have seen gross differences between small intestine and large intestine? What is the difference? Mesentery is attached to small intestine. What about large intestine? Mesentery is not attached. Okay. Mesocolon is attached to, please don't, yes. So small intestine has mesentery, large intestine has mesocolon. So kya farak pada? Okay. Okay. So small intestine entirely what you see is covered with mesentery. That's a fold of peritoneum, right? So fold of peritoneum covering the colon or the large intestine is only to the transverse colons and sigmoid colon, right? But sigmoid colon tak hamara itna dissection removed specimens mein nahi dikhta hai. What else? Tinea coli are longitudinal folds of longitudinal muscles. Okay. They are present at 3 places. So they are present in large intestine, not present in small intestine. So what is another cardinal feature? Saculations or hostrations. What are saculations and hostrations? Okay. The longitudinal muscles which we mentioned earlier, the tinea coli are smaller. Right? हमारे पास कपड़ा बड़ा है, that is intestines बड़ा है, और उसको stitch छोटा सा ही देना है. So what we do, we pleat it. Okay? I usually give comparison with some blouses or some sleeves which we have puffed sleeves. तो वो puffed sleeves क्यों है? कपड़ा ज्यादा है, and the stitch what we are giving is by pleating. तो वो puffs आते हैं. Something similar, the stitch is representing our tinea coli. 3 longitudinal muscle folds है, जिसकी वजह से वो intestine को हमने fold किया है. तो obviously अगर उसमें dilate हुआ वो, तो वो bulge होगा, right? So that bulgings are called as saculations and hostration. One is a gross anatomy term, another is a radiological term. Saculations and hostration. So that's a second cardinal feature. Third, appendices epiploicae. Now these appendices epiploicae are fat-filled peritoneal pouches, right? So 3 cardinal features which are present grossly. This everyone should be able to speak when you have the specimens in the exam. Practical mein identifying small intestine and large intestine is very important. And the first and foremost question asked to you, even though it has been repeated in your schools and colleges earlier also, is the same. The 3 cardinal features that you are supposed to mention, and you mention them as tinea coli, the longitudinal muscle band which is present. You speak about hostrations or saculations which are present, which arise because of tinea coli, and appendices epiploicae, which are present in large intestine but absent in small intestine, right? So, in small intestine, we are going to see the 3 parts of small intestine. What are the 3 parts? Jejunum and ileum, right? And large intestine will be the entire colon, that is starting from cecum to ascending, transverse, descending, sigmoid. They will have same features, right? Is this picture familiar? This is very, 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 very important again. In your microanatomy, right from any slide that you get of alimentary system, not GI system. Difference between alimentary and gastrointestinal tract? When I say gastrointestinal, नाम खुद बताता है, वो stomach से लेके है. Gastro and intestinal. But esophagus नहीं आता है उसमें. So I would rather speak about alimentary system, right? So elementary system में, तो ये this general structure of would be elementary system, but oral cavity and all are little different. So that's why we have used this as GIT. So एग्जाम में कभी भी तुमको माइक्रोएनाटॉमी ऑफ ये जीआई ट्रैक आएगा तो तुमको पूछते हैं 4 लेयर्स के फीचर्स बताओ. ओके, दिस इज प्रैक्टिकल ओरल क्वेश्चंस, दैट स्लाइड वाइवा में. ओके, so there you speak about these from inside out. That's the lumen. I hope you can understand. That's the coil, that's the lumen. And from inside out we have the mucosa, then there is submucosa, then is muscularis externa, and we have the serosa. Serosa or adventitia. What is the difference between serosa and adventitia? Peritoneum. If it is covered by peritoneum, we call it as serosa. If it is not, we call it as adventitia. Either ways, there is connective tissue which is present, there will be lymphatics, there will be blood vessels. But if I am talking about serosa, मतलब peritoneum, तो उसमें epithelium रहेगा. कौन सा? Mesothelium. Or the peritoneum का, that is simple squamous epithelium. Simple squamous epithelium is the mesothelium. This you have to speak. उधर दिखता नहीं है slide में. Higher magnification you will be, but this is what you are supposed to speak. So if you are talking about an organ which is not covered by peritoneum, still that 4th layer will be present, but we call it as adventitia, where only connective tissue and blood vessels will be present. No epithelium, right? Can someone speak about mucosa or mucous membrane as it is called as? Anyone? What are the features there? Since you know a little bit, I want to start. You want to speak? Someone? Yes? Speak it, even if it is wrong, it is okay. We will learn. Abhi kaisa pin drop silence ho gaya. Mucosa, bolo. You have learned this earlier, right? हाँ, तो म्यूकोसा में क्या दिखता है? इधर लिखा भी है. तो बोलो क्या है? Don't speak in chorus. Please raise your hand. You want to speak? So there is mucous membrane or mucosa. So it consists of mucous epithelium. What do you understand by mucous epithelium? Is it always single layered of cells? It may not. So it will vary in the GI tract depending upon where it is. GI tract का main function क्या है? Absorption and secretion, right? So based on that, that epithelium will be modified, right? What is lamina propria? It's a loose connective tissue, okay? What is muscularis mucosa? It's a muscle coat which is present. So when we are talking about this whitish thing, what you are seeing here, smooth thing is the lamina propria. These folds, what you are seeing, will be having the epithelium, and that's the muscularis, right? Outside that, we get to see submucosa. And in submucosa, we get to see glands at times, okay? And we get to see the ducts which are present. And along with that, we also get to see the submucosal plexus. What are these plexus? Which system? Which nervous system? Enteric nerves which are present, they will be intermingling there. So it is autonomic nervous system, right? So that is where we get to see in the submucosa. What are these plexus called as? Submucosal plexus is of Meissner's, M-E-I-S-N-N-E-R, okay, Meissner's plexus. Then we have what is called as the muscularis mucosa, which is inner longitudinal and outer inner circular and outer longitudinal. And there we will get to see the myenteric plexus, right? Again, autonomic nervous system. So this is all richly innervated. What if these nerves are not present developmentally? Is it going to affect? How? Peristalsis will be affected. Secretions of the gland. So basically, such an intestine would be swollen. Okay? You have heard about congenital megacolon, Hirschsprung's. I'll show the picture. So if these plexuses are not absent in certain places, that part of the intestine will be dilated. Obviously, it is not going to convey the peristaltic movement, so it will remain as such. Okay? So it is going to affect the, the digestion process of such an individual, and such a person will come to us earlier. So it will be congenitally affected, right? So that is related to this. Also, since it is autonomic nervous system, emotionally also GI tract is related and affected by stress. Exam hota hai, stress hota hai, gadbad hota hai, right? So that's related to this, right? And serosa we have already spoken about, or adventitia, right? So this is general feature. Based on this, there are few slides where we will be speaking in general about GI tract. So as I mentioned earlier, we have protective, secretory, absorptive and protective functions of the entire gastrointestinal tract. If I say protective, this is seen in esophagus, where the epithelium obviously will be not single layered, but it will be stratified epithelium. So it's stratified, squamous, non-keratinized, right? Exam में आपका ये correlate करना important रहता है, but it is unfortunate that majority of you don't even open your mouth. Okay, this is very simple to understand कि esophagus है, उसको सिर्फ convey करना है food bolus. Okay, so there will be this stratified epithelium, not seen in this image, but I hope you understand. When I see secretory, it will be seen in stomach in the form of columnar epithelium, right? When I say absorptive, there will be columnar as well, because this columnar epithelium is the one which can absorb also. There will be additional features whereby the increasing, the absorption can be increased by various factors, which we will see soon. And large intestine, it absorbs as well as it has to have protective, or the stools are already formed there. So we need to just lubricate it. सो म्यूकस होना चाहिए. म्यूकस कौन सिक्रीट करता है? तो गॉबलेट सेल्स. So it's a common sense कि goblet cells का number will increase as you proceed in the gastrointestinal tract. Okay? This we all know. So small intestine, we are speaking about duodenum. That is a C-shaped structure. How much centimeter? 25 centimeter. Duodeca, दो हाथ के distance का, right? So that's duodenum. When we speak about jejunum, that's the proximal part. Ileum, that's the distal part till the ileocecal junction. And then we have the cecum and all. So total, if you get to see, it is how much? Approximately 6 meters. Okay? So that 6 meter length is the one which attributes to the absorption. So ये जो absorption हो रहा है small intestine में, ये किस वजह से हो रहा है? तो एक तो length है, right? There are other things which are present. I hope you have heard about plica. Plica circularis. What do you mean by plica? Is a fold. Okay? Also called as fold or walls of Kirkring. Or also called as folds, circular folds. Plica circularis. Okay? So that's folds is what you get to see. So this is a gross structure. Okay? आप naked eye से देख सकते हो. अगर intestine specimen देखा, उसको cut किया, तो आपको ये folds दिखेंगे. Either you get to see a complete fold or you get to see a partly fold. Okay, so plica circularis मतलब complete circle, plica semicircularis मतलब incomplete circle. Common sense, right? तो अगर ये gross है तो microanatomy में उसमें क्या है? तो सब म्यूकोजा के साथ वाला fold है वो, right? But microanatomy अगर मैंने slide में देखा तो वो बहुत microscopic दिखता है, उसमें बहुत grossly मेरे को दिखेगा कि ये plica है. You cannot make out. मैं हर पूरा fold को भी बोल सकती हूँ कि ये पूरा plica है. Okay? So naked eye you get to see plica. Microscopically, in this plica circularis, if you see under microscope, you get to see that there are villi, okay, which we have already spoken about. So if plica circularis is present, it increases the absorption surface area to maybe 2 to 3 folds. This villi will increase to 10-fold. और हर villi के ऊपर, you can see this tiny villi, one projection. In this projection, we have seen many cells, the columnar cells which we spoke about. हर सेल के ऊपर उसका प्लाज्मा मेम्ब्रेन फोल्डेड है, तो वो और 20 टाइम्स बढ़ाता है. ओके, सो द एंटायर सरफेस एरिया फॉर अवेलेबल फॉर अब्जॉर्प्शन इज अप्रोक्सीमेटली 200 मीटर स्क्वायर. Such a huge area is provided by these modifications. So modifications or the structures which increases the surface area are plica. Okay, plica circularis or circular folds or valves of Kirkring. I'm sorry, the K is missing here. Villi, microvilli, and crypts of Libercoon. What are Libercoon? Crypts of Libercoon? If there is a villi, the depression, what you get to see next is the crypt. Basically, that's an intestinal gland. We will see what that is. So, plica you have all seen. It is absent in the initial part of the duodenum and in the lower part of the rectum anal canal. The rest of the entire small intestine and small intestine, it will be present, right? So it is present, as you can see, large intestine, it is not that obvious, but it is present more so in the jejunum. You get to see many multiple folds, okay? Grossly, I'm talking about gross. अगर किसी ने पूछा माइक्रोस्कोपिकली वो क्या है, तो माइक्रोस्कोपिकली वो म्यूकस मेम्ब्रेन के साथ सब म्यूकोसा का भी फोल्ड है, सब म्यूकोसा भी उस फोल्ड में चला जाता है, right? So this is what you get to see the folds. So you can see in each of these folds. तो ये पूरा जो है, this entire thing is, but हम जब भी माइक्रोस्कोप में देखते हैं तो हमको ये partly दिखता है, right? So we will be seeing those villi. So that's the circular folds which are present in the small intestine, more prominently seen in the jejunum, right? Everyone with me? Okay. Villi, as we have mentioned, this is the villi, and the depression following next is the crypt, right? If you see villi, what does the villi contain? Obviously, it will have the lining epithelium and it will have the lamina propria. Submucosa nahi aata hai usme. You can see, ye lamina propria mein main structure which is present here are the blood vessels, and there is a lymphatic vessel. We call it as lacteal, right? So that is what is present here. Okay. So the lymphatic vessel, the blood vessels, and the lining epithelium. So lamina propria, along with that, this elevation is what is called as a villi. and the continuation of it where it dips down closer to the muscularis mucosae is the one which is called as crypt. Okay, so we call that crypt area as the intestinal gland, and these are simple tubular glands. Okay, so here you can see, अगर मैंने ऊपर से देखा, okay, not that magnification, these are villi. So if you take a section, you may get to see something like this, and the crypt, when viewed from surface, this will be seen as just a depression. But if you are seeing a section as a whole, you get to see the lining epithelium as well, right? So हमको हर section में इतना clear cut सारे villi और crypt नहीं मिलेंगे. हो सकता है किधर किधर हम लोग को उसका अलग अलग cut section मिलता है. So what is important for us is to understand कि अगर मैंने microanatomy picture देखा, so I should know that this is the crypt. So if this is the villi and the crypt. So if I'm talking about it, this is the villi, right? So if I'm drawing the cells, this is the basement membrane. So cells will be like this, right? और यहां से जब ये आके इधर चला जाता है, so the cells will be still like this, right? What is important for you to know, मैंने जैसे बोला कि तुम्हारे माइक्रोस्कोप के स्लाइड में जब भी तुम लोग देखते हो, तभी वो सेम zigzag पैटर्न दिखता है, but you get to see many other cut sections. You get to see something like round sections as well. These are sections of crypts and villi. Exam में आपको ये draw करने को बोलते हैं. This and this. I hope you are understanding what I am trying to say. When we draw flower, drawing में, how we draw flower? एक गोल draw करते हैं. ये crypt का villi किसका section है? Are you understanding what I'm saying? Is it matching with this or that? Yes. The nucleus will be here. Okay? Obviously, center में core रहेगा. Lamina propria का और वो lymphatics वगैरह जो दिख रहा है. That L stands for lymphatics. This is villi का cut section. Okay? That's a crypt का cut section. See the nuclei where it is? This will be inside. Something like those acini which we draw. The nuclei is here. Can you make out the difference? In your exam, they ask you to draw. When you slide viva के लिए examiner आता है तो crypt और villi का section. So I hope you understand common sense है, but then you look there to the examiner कि कुछ नहीं समझा करके. Okay? Because what is important for us to understand is such sections you get to see multiple times in your slides. So you should know how a villi will be appearing and how a crypt would be appearing in their sections. Okay? तो ये normal जो full draw किया हम लोग ने, that is our villi. उल्टा फूल जो अंदर के साइड में है, acini के जैसा दिखता है, that is the crypt. Everyone clear? Microvilli we have already said कि if you see this entire villi में का एक cell लो और एक cell को देखो तो उसका luminal modification, apical modification is where we get to see the folds of plasma membrane. It has microfilaments in it. If you see structurally, I hope you remember your first lecture when we had done with epithelium. Okay? And there we had spoken about this. I hope you remember the junctional complexes as well. So how each individual cell is attached with the other cell, neighboring cell. Tight junctions are there, desmosomes are there, right? Intercellular contacts. Loose motions, diarrhea जब भी होता है. So the bacteria, normal flora के अलावा अगर कोई viral infection होता है या bacterial infection होता है, in the lumen, that will affect these tight junctions, it will break that junction. So there will be inflammation, inflammatory reaction, as well as these cells lose their connectivity. They are just shed off, right? So that's the basis for you to know that how these viruses and bacteria are affecting. Is that clear? So we have all these things and we know now that surface area is increased, all because of presence of plica circularis, all because of the villi, microvilli and so on, right? Now cells. I know everyone is aware that the cells of the small intestine are called as enterocytes, right? तो काफी सारे सेल्स एंटेरोसाइट्स है, तो ये विला है, ये क्रिप्ट है, बट क्रिप्ट में देर आर सर्टेन सेल्स विच आर न्यू और समथिंग which are named differently and have a specific function. Do you know about them? Yes? Exam में ये भी पूछते हैं. The cells which are present in the small intestine or cells which are present lining the gut. So when we talk about this, we should know that there is an enterocyte which we have already spoken about. Okay? A columnar cell with a basal nuclei. Luminal or apically, there is the microvilli which is present. There are tight junctions which are present. These are multiple enterocytes, how they are adhering to each other with based on the different junctional complexes which are present. Okay, and that's the basement membrane, how they are attached to it. Right? This we already know. So majority of them are enterocyte, columnar cells. Right? Goblet cells, are they present in stomach? Why not? We don't require mucus there. Okay, their stomach requirement is acid. So we have those acid secretion cells, right? But here, as we go on to the duodenum onwards, we require this because we require to lubricate the contents of the intestine. तो इसीलिए goblet cells का number duodenum से थोड़ा थोड़ा चालू होके ileum में काफी रहेगा, okay? Large intestine में बहुत सारा रहेगा, right? So these are goblet cells which are present, and we all know goblet cells are called goblet because what is goblet? Glass. Okay, not that wine glass. Wine glass is different. This is goblet. So if we stain our slide where there are cells containing goblet, HNE stain से हमारा ये जो goblet cell है, इसमें major is Golgi apparatus. Nucleus is at the base. Okay, and these are all those mucinogen or mucus cells which are present, secretions. So these secretions get washed off. There are microvilli which are present, but we don't get to see that, and what we get to see is this, right? We get to see this empty looking cell. You can see this empty looking cell. This is also empty. ये normal cell है, ये भी empty है, empty है, empty. But if you see at their bases, there is nuclei which is present. So in HNE stain, when you get to see an empty looking cell, that doesn't mean the cell is empty. The cell is very much there, but uska jo secretions hai major, wo wash off ho gaya hai. Okay? So those are called as goblet, which appears like a goblet glass. That's why it is called as goblet cells. Right? I hope you should be able to tell the examiner why it is called so. At the base, where the crypt is present, we get to see certain other modified cells. You can see ये भी columnar है. How do I know this is columnar? Based on the nuclei. मेरे को cell का margin तो नहीं दिख रहा है, right? But ये crypt का base है. ये सारे columnar cells है, but ये कुछ columnar cells अलग है क्योंकि यहाँ पे अलग staining हुआ है. Okay? There are these cells with some granules which are present. The granules which are present are zymogen granules. The cells are called as Paneth cells. Functions. Zymogens क्या करेगा? Immunity. If there is any attack, any bacteria other than that normal floral bacteria, okay, this is going to release its secretion, and this zymogen has lysosomal actions. It will kill that bacteria. Okay, so that forms one of the defense mechanisms. So that's what Paneth cell or zymogen cells are all about. You get to see them in special stains. You can see special stain where you get to see. This is some experiment which was done in that mouse cells where you get to see that these are the zymogen secreting cells, and here the bacteria was introduced into it, and you can see they are released. The cells no longer have their zymogen. Okay? Are you understanding what I'm saying? So they are called as Paneth cells. Then we have enteroendocrine or argentafin cells. What are enteroendocrine cells? It secretes hormones. How does it function? Why it is called argentafin cells? It stains with silver stains. Isliye silver is argentafin. That's why it is called as. There are two types. One is the one which is, we call it as the open type, and this is the closed type. So if these are the other cells, and this is the enteroendocrine, also called as enterochromaffin cells. Okay? So they can release their secretion, and you can see the secretion is released, hormonal secretion released into the blood capillaries here. And this is closed. Okay? So these are two types which are seen. No need to mention those types. This is where we can see them that they are stained with silver stain, those black spots. अभी ये कचरा नहीं है slide में का. This is silver stain. Where you get to see that this is what villi, that's the crypt. Slide आड़ा है. Okay? And you can see those black spots. We can see goblet cells also. I hope you can appreciate them. Right? So if you see your slide, identify the cells based on their nuclei. ये सारे nuclei दिख रहे हैं आपको? नीचे? So see their shape and identify the nuclei based on that. Those are goblet cells and these are certain cells which we call it as Paneth. Now in your slides that you are going to see, don't go and ask your teacher there कि हमको ये Paneth cells दिखाओ. HNE में कुछ भी नहीं दिखेगा. सिर्फ तुमको columnar cells वो भी अगर तुमको दिखता रहेगा तो दिखेगा. Majority of you don't even identify that. So columnar cells is what you are going to identify. Examiner coming to your table and asking you questions, you will be speaking all those. Right? So other than the enterocytes, we get to see Paneth cells, we get to see the endocrine cells, also called as argentaffin cells, also called as enterochromaffin cells. Okay? Then we have the M cell. M cell stands for microfold cells because it has a fold there at its base and it lodges the immune system cells. So it is also called as follicle associated epithelial cells. So these M cells are also seen in the crypts. Right? ये may be 2 mark का question आ सकता है. Enumerate the cells which are present in the gut. Okay? So there you speak about all these. Then there are intermediate or undifferentiated cells. Basically these are stem cells. I hope you know that intestinal cells are, there is a turnover rate. ये जो cells अभी है वो 2-3 दिन बाद में निकल जाएंगे. New cells are formed. So there should be some source for these new cells to get formed. So we have the stem cells which will give rise to all types of these cells. Right? So that's intermediate or undifferentiated cells, also referred to as stem cells. Now having known all this, now let's come to our topic and where we speak about microanatomy of duodenum. Classical feature duodenum ka, anyone? Brunner's glands. So here we get to see, that's how the slide will look like. This is a very nice slide. Our slides may not look like this to you, but see it under the white, that binocular microscope, you do get to see such slide. So we know that there are 4 layers which are present. Whatever we have mentioned earlier, all these aspects will be present in this slide. Only difference is we get to see, as he mentioned, Brunner's gland. Now what are these Brunner's gland? These are submucosal glands or duodenal glands which are present in the submucosa. You can see this submucosa all filled with glands. This is the classical feature of duodenum. अगर duodenum बात करते वक्त आपने Brunner's gland के बारे में नहीं बात किया, 0. या draw करते वक्त you don't draw this, you get a big 0. So that's where you get to see Brunner's gland. They ask you what type of glands they are. So you have to say this is compound tubulo-alveolar glands. It is compound tubulo-alveolar. मतलब वो tube के जैसा and there is a coiled part. So common sense, if this is a part of a gland that you are seeing, इसका duct cross करेगा ऊपर. The secretions has to be released into the lumen. So obviously the muscularis mucosae, what you get to see, उसके ऊपर का, will not be continuous. It will be breached. Right? So that is one more feature. So rest all remains the same like a small intestine. We get villi, we get crypts. Lining epithelium is simple squamous. There will be lamina propria, muscularis mucosae which is breached. Submucosa which contains Brunner's gland, also called as duodenal glands, also called as the compound tubulo-alveolar type of glands. And muscularis externa remains the same, inner circular, outer longitudinal. Is that clear? Okay, is it boring? It is, I suppose. So, this is what you get to see in the slide of duodenum. Right? Have I missed out to anything? Yes. Why these Brunner's glands are present in duodenum? Why you want alkaline? Stomach has acid secretion. Acid का काम हो गया. जो भी food chyme के साथ करने का था हो गया. Now it has to move to the duodenum. When it comes to the duodenum should not get, the mucosa should not get affected with it. So duodenum secretes through its Brunner's gland the secretions which are alkaline in nature and also has bicarbonates in it and also has certain enzymes or which will activate the pancreatic juice. Okay, so that is what we are supposed to. So ये खुद का lysis होने के लिए, बचाने के लिए, this is what we are supposed to speak. So there are Brunner's gland which are present and plica circularis, circular folds. Muscularis externa will remain the same and adventitia also remains the same. As we all know, duodenum partly peritoneal. So that's why we speak about the serosa and adventitia difference like this. Is that clear? So that's all about this duodenum. This is how you will be drawing this slide. So these are the villi, those are crypts. This is what I'm speaking about. जब भी हम लोग slide देखते हैं, तो you get to see many cut sections. So you are supposed to identify, because हर slide straight to straight नहीं जाएगा. कोई विला ऐसा आड़ा आता रहेगा तो वो भी cut हो जाएगा. Right? So you get to see those sections as well. Here we get to see the intestinal glands, and these are where, or the crypts rather. That's muscularis mucosae. And this is where we get to see the submucosal glands or Brunner's gland. These are mucous glands basically. So that's how you get to see. And that's muscularis externa. This is the schematic diagram. Draw kiya hua hai. This is the actual diagram. Where this is muscularis externa and that's the luminal side. Right? This is how the glands will appear to. This entire chunk. I hope it is clear. Another slide for you to identify. This part, whenever you get to see glands, blindly call it as duodenum. Okay? There will be no other slide with columnar epithelium having glands in the submucosa. There will be maybe lymphoid tissue. Dot appearance रहेगा. ये glands है. Okay? So submucosa containing glands, it is duodenum. We usually say अंधा आदमी भी देख के बोल सकता है कि ये duodenum है. तो तुम लोग गलती नहीं कर सकते इसके लिए. Jejunum and ileum. Is there a difference between jejunum and ileum? You have seen this commonly asked in your exam grossly. Gross difference between jejunum and ileum. What is the difference? Anyone? Where? Jejunum. So this is jejunum. Few arcades. What are arcades? So we all know mesentery है jejunum और ileum में. Okay. Apart from the difference between the caliber, wall and all, when we see the mesentery दोनों का same रहेगा, right? Thick, lengthwise. Okay. Tube एक side में रहेगा, and that's the mesenteric border where mesentery is present. The yellow thing, what you are seeing, right? Now we have blood vessels which are present. Now blood vessels, when they go in the mesentery, in jejunum they form one or two arcs. You can see एक arc और ये maybe दूसरा arc. And then we have the longitudinal vessels which are running to the mesenteric border of the intestine. So they are called as vasa recta. Vasa मतलब blood vessel, recta is straight running vessels. तो अगर vasa recta longer है और arcades छोटा है तो वो jejunum है. तो obviously यहाँ का जो windows है, what do you mean by windows? Between two vasa recta, the area that you get to see. तो अगर vasa recta लंबा है तो वो विंडोज रेक्टेंगुलर रहेगा, लंबा रहेगा. It won't be squarish, right? Here arcades are more. You can see एक arc, दोन arc, 3 arc, 4 arc. Something like you remember कि arcs जो है blood vessels का jejunum से लेके ileum तक बढ़ेगा. तो एक है, फिर 2 बनेगा, फिर 3 बनेगा and 4 बनेगा. तो obviously vasa recta जो longer है and windows जो rectangular है, आगे जाके vasa recta will become smaller and windows will become squarish. Okay, that is what you are supposed to remember. Okay, and here the windows, when we speak about windows are because of the fact the windows may not be seen so clearly, but this is what you are supposed to speak about the arcades and vasa recta. Right? This is about gross, which you should know. When we speak about microanatomy, now too many things here. Importantly, जो हमारे 4 layers है, वो 4 layers चारों के चारों वैसे ही है. Lining epithelium is columnar. As you can see, there will be few of goblet cells which are appearing now. ज़्यादा नहीं है, लेकिन है, right? Glands, intestinal glands वैसी है. बीच में you may get to see some lymphatic nodule which is present. Okay? एक हाथ दूसरा lymphatic nodule, not much. Muscularis mucosae वैसी ही है. सब म्यूकोजा वैसे ही है, कुछ नहीं है उसमें खास. And muscularis externa भी something similar. So with circular muscle and longitudinal muscles, right? Nothing great. So that is what is jejunum. Okay? But the villi which we get to see. Now when we speak about the villi will have what is called a striated border. Now what is striated border? Because of, when I say striated border, हमने क्या बोला था? अपाइकल सरफेस में क्या होता है हर इंटेस्टाइनल सेल के? माइक्रोविलाई. Now this microvilli may be regularly arranged, so we call it as striated. Because जब भी उसको देखते हैं तो वो कुछ तो लगता है कि कुछ तो है ऊपर. तो the appearance is not smooth, but it is striated. So microvilli, which is regularly arranged, gives striated appearance. This is jejunum. Microvilli, which is not, what do you say, regularly arranged, okay, they give rise to what is called as, in the kidneys we get to see this. Okay, we get to see brush border. Now many books give you कि ये जो striated and brush border है वो same है. Okay, but when I say brush border, I speak that this regular arrangement is so regular that they appear straight. और यहाँ पे striated दिखता है क्योंकि थोड़ा सा ऊपर नीचे है. समझ में आ रहा है? Brush border, which is seen in kidneys, to be very specific, proximal convoluted tubule (PCT). वहां पे ब्रश बॉर्डर होता है क्योंकि वो जो माइक्रोविला है वो एक समान है, स्ट्रेट, और यहाँ पे वो स्ट्राइटेड है. आजकल हमारे ब्रशेस भी स्ट्राइटेड होते हैं, बट रिमेंबर दोस अर्लियर ब्रशेस विच आर स्ट्रेट ब्रश, टूथब्रश, उसका बॉर्डर कैसा है? ब्रिसल्स का सेम लेंथ का है. ओके, तो सेम माइक्रोविला और ऑफ सेम ये, एंड दे आर रेगुलरली यूनिफॉर्मली अरेंज विद द सेम लेंथ. सो दैट्स ब्रश बॉर्डर. दिस इज स्ट्राइटेड बॉर्डर. सो माइक्रोविला आर नॉट रेगुलर, दे आर इरेगुलर. राइट? इज दैट क्लियर? Yeah, second thing है which examiners may ask you, what do you mean by striated border and brush border? So striated border and brush border are both because of microvilli. If this striated border is seen in intestine, specifically more so in jejunum, because these microvilli are not so regularly or uniformly arranged, that's why they give striated appearance. When they are regularly arranged, they are seen in kidneys, PCT, which we say as brush border appearance. Okay? The villi here, they are called as tongue shaped. Now books में बहुत सारा लिखा है, डिओडिनम का कुछ लीफ शेप है, ये ब्रश शेप है, कोई टूथ शेप है. You follow whatever you want to, क्योंकि मेरे को उधर कुछ नहीं दिखता है वैसा villi. Okay? You write in your exams also if you understand it better. But we identify duodenum, jejunum and ileum based on the features. ये जो शेप है वो वेरी करेगा obviously. Okay? But आपको अगर आपके बुक में देख के लिखना है अलग अलग शेप तो you can write. Okay? Submucosa will not have anything specific as is mentioned here. Muscularis externa will remain the same and serosa will remain the same. So that's jejunum. तो है क्या इसमें? तो इसमें सिर्फ goblet cells है, और अगर होगा तो कुछ lymphatic nodule होगा, वो भी नहीं हो सकता है. Right? This is what jejunum would be. You can see many goblet cells now. दिख रहा है काफी सारा. I hope you can appreciate them. Right? जो भी empty looking है, and you see in between the cells, you get to see those columnar cells typically. Okay, and these are those intestinal glands, the crypts which we get to see, right? The rest remains the same. You can see partly here, the lower half, right? Ileum, you get to see the classic eye-catching thing is the lymphatic nodules that you get to see. They are called as Peyer's patches. Again, books have difference in opinion. Now these Peyer's patches are actually present in the submucosa, and they may extend into lamina propria, or they are present in lamina propria and extend into submucosa. You write whatever, but mention that they are present all throughout. They are lymphoid aggregation, or they may come together and form what are called as patches. So they are, or nodules, they are called as Peyer's patch, right? So this is what you get to see. Rest features remain the same. Wherever these follicles are present, we get to see in the crypts those M cells which are present. Remember microfold cells? क्योंकि lymphoid follicle मतलब क्या? It is related to the defense. M वो microfold वाला सेल क्या कर रहा था? वो लॉन्च कर रहा था, right? Immune system का. तो obviously इसके ऊपर जो भी crypt रहेगा उसमें काफी सारे M cells रहेंगे. उधर स्लाइड जाके ढूंढो मत M cells की तरह. M cell नहीं दिखेगा. Okay? So these are all theory questions which will be asked in your microanatomy practicals, right? So that is what you get to see here. Are there any questions, queries? number of goblet cells is now going to increase because we are proceeding, right? So that is what you get to see there. Now this is one more term. I hope you know this. Malt, galt, balt, whatever, different things. This is malt. Malt means mucosa associated lymphoid tissue or lymphatic tissue, whatever you call it as. Under these, we get to see different, it can be the conjunctiva associated lymphatic tissue. N stands for nasopharynx associated lymphoid tissue. B stands for the bronchi associated lymphoid tissue. This is gut associated. So अभी हम लोग जो भी बात कर रहे हैं वो galt है, right? So if asked, they come under this particular group. And V stands for maybe vaginal related or the vesicle related. Vesico-vaginal associated lymphoid tissue. Oh, guessing those? Okay. So that's malt. As a group, broad name, under these things would be there. Right? ये PG level में usually question आता है उसके under and you have to describe. आपके लिए maybe just knowing what is malt, galt, you should be aware about it. Okay? These are the differences between jejunum and ileum. As such, very small picture, can't identify. Apart from Peyer's patches, we see the length of the villi. The villi length will be smaller in ileum. छोटे छोटे हो जाते हैं, right? M cells तो दिखने वाला नहीं है, Peyer's patches दिखेगा, right? See, it is present extent throughout the lamina propria and submucosa. Is that clear? Everyone with me? Those who are awake, sorry. This is the difference. I'll be sharing this PPT so that you need not go searching for it. So these are the basic differences between duodenum, jejunum and ileum with respect to the microanatomy part. So this is what we are supposed to know. So epithelium we all know, columnar है, columnar striated है, goblet cells बढ़ जाता है, यहाँ पे few था, +1, +2. Villi, as they have mentioned here, you can see different shapes are mentioned. I said I will not rely on this, but villi will have lesser height as we move ahead. Lamina propria, crypts are present in all. There are no Peyer's patches in duodenum, but lymphocytes and all aggregations are present in jejunum and Peyer's patches which are present so in ileum. Brunner's gland is the classical feature that is present in the submucosa of duodenum, and rest there is nothing except for Peyer's patches in the ileum, right? ये दिमाग में आपको छपके रखना चाहिए ताकि अगर कोई पूछता है और whenever you see slide, we do differential diagnosis. What I mean by differential diagnosis, I see this and this. I know कि ये और यही slide हो सकता है. Rest नहीं हो सकता है. Okay? I see 4 layers. I cannot say this slide is that of maybe a lung. Lung में कुछ layers है ही नहीं. Okay? तो इतना grossly mistake मत करो. So this you should be able to speak about. Gluten, allergy to gluten. So that's celiac disease or celiac sprue. The villi are lost in this because of allergy to gluten, whereby this villi will become totally lost and this person will have obviously absorption related disorders. So we'll present to you with this, where you get to see that a biopsy will reveal that there is no villi which are present. So this is celiac disease. That's small intestine and that's large intestine. What difference do you see? See the villi? No villi here. But crypts would be present, right? These are just folds which are seen. So there will be crypts which are present. So don't ever mention villi ka naam in intestine. Colon mein villi nahi hote hai. There are just crypts which are present. And when you see the muscularis externa, even though it is not so clearly seen, hai, thoda bada hai. Okay, that's bigger here. And we get to see what do you call as, see this myenteric and Meissner's plexus, ऐसा हमको उधर microanatomy में नहीं दिखेगा. This is what you are supposed to speak about. We get to see tinea coli का वो aggregations दिखता है colon में. Okay, if that section has gone through it clearly, you get to see those aggregation maybe in a bigger form. Okay, we will see in certain slides. So this is what we had seen, lymphoid aggregation, the Peyer's patches which are present here. And later on we do get to see, but again in form of little lymphoid aggregation. Okay. In the duodenum there was Brunner's. Later on in jejunum there was nothing. In ileum we get to see Peyer's patch. As We move ahead in the intestine. The 4 layers remain the same. Only thing is mucous membrane doesn't have villi. There are just crypts which are present. The lamina propria and the muscularis mucosae remains the same. Submucosa will have some lymphatic nodules. That muscularis externa remain the same with an additional maybe an collection of longitudinal muscles which may be seen. Okay. So colon, we have already mentioned the parts of the colon. That's, you can read what you get to see in mucosa. Epithelium remains the same, simple columnar. Multiple goblet cells will be seen. The glands are there, the dipping down of the crypts, that is the simple tubular glands. Submucosa will not have anything, no Brunner's glands, no Peyer's patches. I'm sorry, uska orientation thoda hai. This should have come earlier. Okay. So 3 bands of Taenia coli would be seen, and serosa will have a connective tissue layer which is present. So colon do not have plica circularis. So grossly you will not be able to see those folds. Microanatomically you will not be able to see villi at this magnification, and higher magnification you won't be able to see the Paneth cells as well. Okay. So abhi ke liye aapke liye sirf villi nahi hai, right? So this is what in general you will be seeing, right? The picture itself is, you can understand. So this is where multiple goblet cells you get to see. That's muscularis mucosae. Submucosa doesn't have, and this is where I was talking about Taenia coli. You can see the longitudinal muscle coat. Obviously longitudinal जा रहा है muscle, इसलिए वो टिपका टिपका रहेगा, dot dots, right? But ये dot dots इधर काफी सारे हैं. हमने पहले भी बोला था Taenia coli is a band, so that band is what you get to see, right? That's Hirschsprung disease, the megacolon, which I spoke about. Absence of Meissner's and Myentrick plexus at certain places. In certain individually may be diagnosed at birth. So it is called as congenital megacolon. Congenital means since birth. Mega means big. Colon is obviously large intestine. Okay. So that part of intestine will not be involved in peristaltic movement. I hope you know peristaltic movement spreads like a wave. तो एक कॉन्ट्रैक्ट हो रहा है, एक डायलेट हो रहा है एंड सो ऑन. अगर एक जगह पे प्लेक्सिस ही नहीं है तो उस पर कोई सेंसेशन ही नहीं रहेगा, द इनर्वेशन ही नहीं रहेगा. सो दैट विल रिमेन एज इट इज. ओके. सो ऑब्वियसली ओवर अ पीरियड इट विल लीड टू डायलेटेशन. Appendix, the vestigial organ present at ileocecal junction. We get to see the 4 layers maintained here. Again there is no villi which is present. There is simple columnar epithelium with numerous goblet cells. Okay. And main is what you get to see is there is numerous lymphatic follicles which extend into the mucosa. The slide is tiny. Okay. If you see on the slide, the tissue which is present is a very small tissue. And that's where you can identify. The muscularis externa is not that great because as compared to the large intestine. This is the slide. That means in one picture, one focus in low power, you can see the entire slide. People come and ask, ये large intestine है या समझ में नहीं आ रहा है या ileum है, I don't make out. Ileum में तो हमको दिखेगा, villi है लेकिन कम है, right? But आपको जो Peyer's patches और इसके जो lymphoid aggregation में confusion होता है, that confusion will not be there because this slide you get to see entire in one, focus, right? I hope you understand what I'm saying, right? So this is where you get to see the lymphoid tissue which is present and the layers that are present, right? Rectum anal canal is not your exam slide, but still this is where you can see the junction of the rectum where the epithelium would be the same, and this is the anal canal where we know anal canal is how long? Centimeter wise? नहीं पढ़ा है. Okay. So anal canal you get to see some 3 to 4 centimeters long where we get to see that there will be stratified keratinized epithelium in the lower part, right? Same as that of the skin. So this is where you get to see the difference between epithelium change. For TRM or your exam, these are the slides that we have. So all these slides can be kept in your exam. But it's a common sense, when we keep slides, we keep from one system at a time. So if you have 10 slides for your final exams, so it would be something like general microanatomy में से 1-2 slides रहेगा, राइट? रेस्पिरेटरी में से एक रहेगा, सो वो कॉमन सेंस यूज करो. फिर भी एग्जाम में जो नहीं रहता है ना स्लाइड लिस्ट में, वही लिख के आते हो. यू हैव दोज लिस्ट 57 स्लाइड्स. बाकी सब तो रट्टा मारते हो, उसको एक बार देख लो. But rather I would say learn these slides, because if you don't know normal, you will not know what is abnormal later, second year. Pathology nahi samjhega. Okay? So when you see the slide, if you are not able to identify the slides under the normal microscope, see the slide under that binocular microscope. Our slides are good. Okay? Try to understand their features through that the binocular microscope and identify the features, make your own notes so that you understand. They are not difficult to understand. You don't put your, I don't know what is the problem. End mein, you know, many a times you don't identify the slide, because these slides jo 10 mark hai aur tumhara viva ka marks hai, it is something like you can get 20 on 20 aaram se. But still there is mistake, and even if you do mistake in identifying in the slide viva jo 2 slides rehta hai, we ask you questions, we bring you back to the bata ke ki ye slide ye nahi hai to aur kya ho sakta hai, and we give you tips. But agar tumhara hai hi nahi kuch differentiating, तो कुछ नहीं हो सकता है, because you don't think. नहीं, don't laugh, because this is seriously, मतलब whenever we give you clues, you don't listen to those clues. Okay, don't उधर पहले से ही प्री नोशन बना के गए ना कि सब ब्लू और पिंक ही दिखता है, तो कुछ नहीं दिखेगा. Try and see and identify, maybe पूछो उधर टीचर को. Okay, या फिर टेक्स्ट बुक में देखो. These slides are very much with you all, right? You people click good images also. I have seen that last time. माइक्रोस्कोप में से निकालते तो उधर पूछो ना कि क्या है, कैसा है. I know it for first time it is difficult, but still not to that extent. You people are all good, तो ये हो जाएगा आराम से, right? In this slide, as such, with this magnification here, because I wanted to show you the entire slide, nothing is seen. To be very frank, everything is pink. Okay? Here at least you can make out these Brunner's glands. यही दिखता है. उसको थोड़ा बाद में zoom करके बाद में देखो, because तुम्हारे identification वाले slide में whenever we make you identify for the spots, तो हम focus करके रखते हैं, तो focus इस पे रहता है. Okay? So, और थोड़ा luminal part भी रहता है. अभी इसमें नहीं आया, but lumen का part रहता है. तो वहां जहां पे भी white दिखता है, you know that is lumen. ये काफी सारे folds है, तो obviously this is villi. You get to see, ये gland है करके तो पता चलता है, और muscle है, तो ये layers 4 layers है, मतलब ये gut है. Okay? This is how you have to differentiate. Not seen so clearly. That's jejunum. This is where you can see. ये दिख रहा है आपको villi के, I would say टुकड़े टुकड़े. This is not टुकड़ा टुकड़ा. This is exactly what you see there. But when we prepare the slide, it is not mandatory कि वो slide एक ही section, because there are multiple villi, right? तो वो जब भी section जाएगा तो आपको बाकी के आजू-बाजू के villi के भी section मिलेंगे. Okay? You see one particular villi, you see that there are many goblet cells. ये दिख रहा है white छोटा-छोटा. These are all goblet cells. And then you see lower part, you don't get to see any classical feature there. So that's jejunum. Okay? See the goblet cells? Now you can make out. Okay? That's ileum. This is the lymphoid tissue. जो तुम लोग schematic diagram तुम लोग draw करते हो, तभी dot dots draw करते हो. So they may be a classical with a follicle, with a germinal center, or they will be just haphazardly arranged. That's again lymphoid follicle here which is present, and those are those crypts which are present. That's large intestine. A thick muscularis externa, nothing much in the submucosa. You may get to see a lymphoid follicle, but plenty of goblet cells. And you see the villi, uska ye, we don't call it as villi, we call it as crypts. Kuch hai hi nahi waisa, right? These are all crypts which are present. No villi, right? You can see all goblet cells, sub-goblet cells. And that's appendix, again those crypts. And you can see the entire slide with the lumen in a same picture. Okay, this is what I'm trying to say. You can see the entire wall. See the wall muscularis externa is very tiny, small here. Okay, and this is all lymphoid follicle. Differentiate करने को आना चाहिए कि lymphoid tissue कैसे दिखता है, glands कैसे दिखता है, बाद में higher magnification पे देखेंगे. तो इसमें कुछ भी as such structure नहीं दिखता है, it's just dotted appearance. Okay, this is what the lymphoid follicle would appear like. Okay, see the goblet cells in the crypts. I am a tubular structure, matlab some tube with a lumen, right? This is how you can remember your slides in your own way. Some people who can do it, I found it, this was very easy to understand. Okay? May not be, sab log ke liye nahi ho sakta hai ye, but still. All members of my family are lined with simple columnar epithelium. Simple columnar, agar tubular structure bola to wo vessel bhi ho sakta hai, but vessel columnar nahi rehta hai, to out. Okay? So this is intestine. Right? Then it is written, I am almost similar to my brother. That means there is some two slides with the same structure. Right? We both do not have villi and goblet cells. When I say villi nahi hai, to ho sakta hai colon bhi hai, but usne bola goblet cells bhi nahi hai. So it is definitely stomach. Clearly stomach pe aa gaye. Then the only thing different between us is our lamina propria. I have glands which are 2/3 secretory and 1/3 conducting. So to be very specific, when I say pylorus or fundus, is this jejunum? Not ileum? Why? Peyer's patch. Correct. Ring of lymphoid tissue in my submucosa, and I do not have intestinal villi. If I say I do not have intestinal villi, that means it is colon. But colon में ring of lymphoid tissue, matlab it is appendix. Appendix has a complete ring of lymphoid tissue. So that is one additional feature which you should mention. Okay? कहाँ पे भी breach नहीं रहता है. That's it. Thank you.